monophyly means you have an ancestor and you have a taxonomic group, a class, a family, a genus, whatever. And everybody put in that group comes from that one ancestor. That's monophyly. Monophyly means one group, one lineage. And so there's our ancestor. Let's just say radiated off. And I'm not drawing a cladogram here. I'm just drawing a mushy evolutionary tree. These are all, let's say these are all species at the top here, okay? These are species A, B, C, D, E, how am I having G? All right. So, this is the phylogeny someone recovers when they do their research. That's the word we use, we recover a phylogeny. So that's the hypothesis, and it's a hypothesis, it's not truth. It's not the final word, but that's our best hypothesis. So we base our classification on that. And that's a monophyletic group, because it comes from one ancestor, and we're not leaving anything out. And for example, suppose someone said, oh, here, this other species over here, H. Let's put that in that genus, or that, not that genus, let's put that in that group. We can't, right? Because his ancestor's over here. He might be super similar, but that doesn't count. He's got a different ancestor. So, this is the monophyletic group. I don't know what that confuses. And those are species. So, and we define the monophyletic group, and give us a little more room down here. We don't have the ancestor. We don't know who that was. We can just hypothesize. You know, we know he existed, and we can form some hypotheses about what he looked like, but we don't have him. But what we have is a group we think are all related, and the reason we think that is how we draw these. We put a bar here, and that represents the character that evolved in this ancestral lineage, and everybody still shows it. That's why we put them together. We say, so there's some feature of their skeleton. Their nasal bones are long and pointy, and that's unique. Okay? Pointy nasal bones. And that is a particular feature that nobody else has in this whatever group it is. It's lizards. So that's a trait that evolved here. Everybody shows it. That's why we put them together. And usually you have several. You like to have confirmation. You don't. Sometimes we just have one. Okay. But you might have 20 traits there, or you might have someone that run some DNA gels on chunks of tissue that someone told him came from these lizards, and he might recover a bunch of DNA sequences, and that would support that as well. So here's a group of 4 species. They have an ancestor too. And what's this trait called? What do we call it in general terms? What do we call that?
Synaptomorphy.
That's a synaptomorphy. Yeah. Shared trait. That's what that means. And so that's the synaptomorphic group. There's another synaptomorphy here. And for these guys, there would be a different synaptomorphy. And there'd be one here, right? And one here. So when you're doing these kind of studies, you look at the whatever data you're gathering, you look at all the features, and you look for the synaptomorphies. And again, we define those, again, the computer algorithm that does this, we define the importance of those synaptomorphies comparing our guys to the outgroup. And that's fraught with a lot of challenges, as you brought up. But it gives us hypotheses we can defend. Now, all such problems here, suppose let's say we've got 6 synaptomorphies down here. That's 6, yeah. And the main one's pointy nasal bones. But suppose this guy doesn't have pointy nasal bones. But he's got the other 5 synaptomorphies. Well, our hypothesis would be his ancestors said, I don't need those pointy nasal bones, and got rid of them. But that's why you like to have lots of characters, lots of traits, big data sets, because you can kind of weed out the reversals or other things that confuse you. But in a very simplistic system, like the one they always show in the textbooks, vertebrates, got a fish here, amphibian, a reptile, a mammal. We can make it very simple. So anyway, so just see, this is a monophyletic group, right? Defined by this trait. Here's another monophyletic group, defined by this trait. Here's another one, defined by this trait. So monophyly is what Willie Hennig wants us to use, and that was his introduction, his concept, and phylogenetic systematists are really big on it. And you know, I was still going to meetings as a graduate student when this stuff, this way of doing things was fairly well established by the late 70s. I started going to meetings in the 80s. But there were a lot of the older guys and gals were fairly resistant to this. And I think it's a pretty useful system, but I find the people who promote this really dogmatic. And in the early years, the 70s and 80s, when I was going to meetings in the 80s, the people promoting this had the reputation of being absolute jackasses because they were so aggressive. And you'd give a talk, and if you didn't use the words right, they would stand up and nail you on a question. And I had one colleague who I got to know later. I'm not going to say his name. He's really, really good ichthyologist, and I like him a lot. But he was one of those early people, and I'm just going to say his name is Bob Smith. It's not. But for years, I never heard the name Bob Smith without the descriptor in front of it, that asshole, Bob Smith. But it's just, he was an early adopter. Sorry for the language. So, we define these monophyletic groups, and in this example here, A, B, C, D. If each of these at the top here is a species, each of these top here species, well look, we have two different groups of species. Two monophyletic groups of species. And our classification system here, this whole thing, because every taxon has to be monophyletic, the whole thing we might rank as a family. And each of these subgroups would be genus. A genus 1 and a genus 2. So everything's monophyletic. The family's monophyletic, the two genera are monophyletic, and every species is monophyletic. A little bit clearer? Okay. Like I said, we can spend a whole semester on this. We're not going to. Okay. Well, let's just carry on. I got a few more things I want to say about classification, and then we'll launch into vertebrates proper. Here's my buttons here. Okay. So we were talking about you identify species and then you give it a name, and I just want to talk about the nomenclature a little bit because it's a big part of what I used to do. Nomenclature is where we're at. That's the giving everything you need. Remember, nomenclature is everything has to have a unique name. Every species has a unique name. And Linnaeus gave us a general way of doing this in the 1700s, but there were no rules. And by the late 1800s, there were enough scientists working that we needed a standard system, not for doing the science, but for applying and changing the names. And gave you some examples as we go along here. So the International Society for Zoology got together, the International Zoological Conference in 1898, and they wrote a set of rules for how the names are made. The year Homo sapiens. Well, there's rules about how that's named. And the first set of rules were in French, which by the late 1800s was the language of science. And that's right there, the international rules for zoological nomenclature. And it was a set of rules by which we would craft the names, not do the science, just apply the names, and change them if they needed to be changed. That was to stabilize things. Remember, classification is at its core about communication. By 1904, we switched to English. Actually, French and English, they were parallel languages at all the meetings and all the publications. And we formed the International Commission for Zoological Nomenclature. And their journal is really, really boring. I used to get it. And the International Commission for Zoological Nomenclature has a set of rules, and I think we're in the 4th edition. I'm not in touch anymore, but I forgot to bring it. I have this in my office. I'm the only person you know who has a copy of this. And it's a set of rules. And the rules deal with things like, what if the same species, the same evolutionary unit, C here, two different scientists from two different countries in two different times gave it a different name. One scientist found it, gave it a name, and then 20 years later, another scientist encounters it again, gives it a name. Which name do we use? We got one evolutionary unit, we got two names. Things like that, what these rules cover. The overriding goal here is that we have stability in nomenclature, because that confuses people like Sean Anderson when it's not stable. You always know Sean, right? You're going to go tell Sean I'm making fun of him. No? We want the nomenclature to be stable, and we achieve that stability when the overriding rules here is called the law of priority. Those are the two fundamental underlying things in the rules. Stability through the law of priority. And basically what the law of priority says is that example I gave you earlier. One scientist described this species 1904, and 20 years later, 1924, another scientist saw it again, and didn't see the first description and gave it another name. Law of priority says we always go with the earliest thing, unless there's some reason we shouldn't, as there's exceptions. But priority is first. So one of the things that gives us stability is what we call the type concept. All taxa must be rooted. What that means is the taxon has a base. And so, for example, when you erect a family, when you create a new family of fishes or lizards or whatever, you have to say what the type genus is. Remember, category family, and then the next category down is genus, right? We're good with that? So here's my new family. I'm going to say, okay, here's the type genus of my family. If there's ever any question about what this family is, it's whatever this genus is. Each genus is rooted by a type species. So if there's ever any question about what fishes or lizards we put in this genus, it's whatever this species is. Here's what species, I list it. And then every species is rooted with a specimen, a type specimen. So if there's ever any question about what that species is, you go look at the specimen, you go look at the type. Doesn't matter what people are saying it is, it matters what the type is. So we've got this rooted concept. Let me give you some examples. Let's start at the bottom with type specimens. And I've described about 25 new fishes. New, that doesn't mean new evolutionarily evolved. That means no one noticed them before and hadn't given them a name. So for every one of the species I describe, I designate types. And so if there's ever any question about what I was talking about when I described Ethios malugoi, a little fish from Mexico, you can look at my description, you can look at photographs, but the real indicator is you go to the University of Michigan, you go to their type safe, you open it up, it's actually a type closet, you take that jar of fish out and go, okay, that's what that name is. Whatever this is, that's what that name is. And so here's some types. There's 3 or 4 different kinds of types. A holotype, that's the single most important specimen. When I describe a species, or a scientist does, and they pick the nicest one that shows the traits very cleanly, if you have a choice of multiple specimens. I usually pick big males when I'm doing fishes. Nice big adult. There's no ambiguity there. And that's the one. So this is a little fish from China, and that's the holotype. If there's ever any question about what that is, you don't look at the paper, you don't talk to your colleagues, you go look at the specimen. So that's the one. And it has to be picked by the person doing the work. So our friends Zhao, Wanatabi, and Zhao, they had to pick the holotype and say, this is it. 100 years later, we can't pick it. It's too late after those guys are gone. And that's preserved in a museum somewhere. Usually in an important museum. You put holotypes places where you know they're going to be kept, like the Smithsonian, the California Academy of Natural Sciences. It goes to a major museum where they're preserved and you know they'll be curated. And anyone who needs to can go see them. So there's a lizard holotype. Here's one I worked with in my African fishes. It's about this big. It's what we call in the business a crispy critter because it's dried out. And I had to figure out what that was. Well, that's Anabas togoensis. I had to figure out what Ernst Dahl was talking about by looking at that specimen. And when you read his description, it was dried out when he had it. He never should have erected a species on that crappy specimen, but he did. And we had to figure out, because that name's in literature, we had to figure out what that was. I did, and I'm still not sure because it's a crappy specimen. Sometimes there's not just one type. Sometimes there's multiple types. And there's a category called syn type. Syn, shared or same. And these, again, these are designated by the original author. The guy or gal doing the work has to pick these. And syn types are when you don't want to make one, like maybe there's a male and a female, and they're different, so you pick syn types. I don't know what the case is with these guys. Oh, that's Ernst Haeckel. That's a long time ago. That's why he used syn types. Sometimes it's from a long time ago. Ernst Haeckel was doing this in the 1840s. He didn't have the holotype concept. It didn't exist yet. He said, here's my types. And there's two of them in the jar. So they have the same rank. So they're like holotypes, except there's more than one. Usually just a few. And I don't think anybody would designate syn types today. It's an old-fashioned thing. But we do this today. Para, when you see para in a chemistry word or a biology word, it means beside or next to. And paratypes, one more time I'm going to say this, these are designated by the original authors. This had to be done by Eccles and Lewis. And these are specimens. These guys, I'm sure, picked a holotype and put it in the British Museum. And then, because we know species are variable, you can't represent a whole species by one specimen, you shouldn't. So they picked another 20 or 30 to get males and females, to get juveniles and adults. They picked another array of specimens and they call them paratypes. There's the one, and then these are the next tier down, they're the extra types. They're paratypes. And usually you do this to represent the geographic range maybe, or males and females, or young to old. And the other thing that might not occur to you because you haven't done this work is when the holotype goes to the Smithsonian or the British Museum, an important museum, and you can take a jar of paratypes and say to the curator, can I send these to the Philadelphia Academy of Natural Sciences, or can I send this jar of paratypes to another museum? You kind of spread them around. And it lets you get your name on the desk of curators at other museums. So that was my perspective on that. But these are secondary types, usually get spread around a little bit. So all of these are designated by the original author. And I'm giving examples. We're coming to examples of why we have all these rules. This next type is called a lectotype. So a species was described. We have types. But it was probably done 100 years ago or 120 years ago, and they're just called types. The author didn't say, here's the one. He just, here's my types, there's 20 of them. But there's no holotype, because he or she didn't pick one. Somebody later comes along and says, you know what, we should really have one type. We should really pick one type and say this is the best one. We can't call it a holotype because the original author didn't do it. So someone else comes later, looks at the types, syn types or paratypes, and says, here's the one. This is the best one in the jar. This is the lectotype. So a lectotype is like a holotype, but it's designated by somebody later. And it's usually done to try to stabilize the nomenclature. Say, let's make sure this name has a good foundation. Neotype, new type. Here's another one. The original author's long gone, or they never designated a type, or they did designate types, but the museum burnt down or was destroyed in World War II. So the types no longer exist, or there never were any. But in order to stabilize the nomenclature, we need to nail this name down. We want a specimen tied to that name. So if there never were any types, or the types are known to have been destroyed, someone who's really sharp and knows this group of whatever really, really well can say, I'm really, really 99% sure what Saville Kent was talking about when he gave this name, when he made this name. 99% sure I know what he's talking about. Here's a really good specimen of that thing. Let's make this the type. Can't be a holotype. We call it a neotype. And only the commission can make a neotype. You can ask the commission to make a neotype and they'll sit and think about it. So like I said, it's all to stabilize the names. And all these things came up because there was some instance where some taxonomists were trying to determine the correct identity and correct name for some taxon. And they ran into a problem, and these things were created to stabilize that. So holo, syn, para, lecto, neo. The last kind, I looked and looked, it's the only one I could find an example of it. The last kind is a really rare one. It's called an iconotype, like an icon. And it's when the type is a drawing or a photograph. And these are really, I looked and looked for one. I actually found a fish one. But there is no specimen that Lassapade that ties this specimen down, or this name down, damn it. But that's a really good drawing. His description's clear enough that's good. And that's rare. In this particular instance, that's the iconotype. My friend Phil Heemstra said, you know what, I don't want an iconotype for that species, and so he picked a neotype. And now we have a neotype. But that stabilized the name. And there you go. That's the animal. Again, this is all about nature. This is all about getting back. That's the evolutionary unit. That's the ecological entity. This is just how we talk about it. Let's find a better picture. When I'm working with all these specimens, I pause every once in a while. Remember, it's not a dead fish in a jar. That's just representing an evolutionary unit in Africa, an ecological entity in African streams. I always try to tie things back to the real thing. And we spent a lot of our lab time in this class with dead things, specimens. We're going to do some anatomy labs. But I never want us to lose sight of the representation of real of living things. It's more and more important every day. So again, just some examples of things that come up here. This fish I used to work with when I was in Arizona. This is a real common, that is one of our common western river minnows. Get about this big in the Colorado or Yampa River, things like that. This is the evolutionary unit, ecological entity. That's our designation for it, Gila robusta. That's the genus and species. That's our current designation. Baird and Gerard described it in 1853. And they described it 2 other times in the same paper. 3 sequential pages, they describe the same species 3 different times. They give the same evolutionary unit 3 different names. That's why we have the rules. And this is a very variable species. Small specimens look different than big specimens, so they thought they had 3 different things. And then actually Gerard came back a little while later and gave it another name. You can put it in a different genus. So these are all names that people have applied to this single, what we now consider to be one evolutionary unit. So which name did we pick? Law of priority says we picked the oldest one. And Gila robusta is described on the page right before Gila gracilis. 1 page. Okay. Just one more example, and I want to move on. But I got 2 examples here. Which one am I going to go with? I'll go with this one because it's enormously complicated. Here's Georges Boulanger. This we used to do in libraries, and when I worked in a museum in Europe, they had little file drawers. Every paper that had ever been published on African fish, they had a copy of it, if not a copy, a photocopy of it, these little drawers. Nowadays you can click and get a lot of this stuff on the computer, but we were trooping back and forth to the library, looking for obscure papers all the time. Georges Boulanger, prominent Belgian ichthyologist, worked with African fishes, and he created these 2 names. It doesn't matter if they're in different genera. He at one point decided that one genus didn't exist, but about 6 years apart, he described 2 different species. Let's say he erected 2 different names, Weeksii and Oxyrhynchus, and there's his types. Weeksii and Oxyrhynchus, those are the types, and so that's what I had to work with. And this was my group of fishes, so I was trying to figure out. I wanted to make sure every evolutionary unit I recognized as I got the specimens out, I wanted to make sure it had the appropriate name next to it. And the appropriate name is what the type is. And there's his description of Weeksii. It's just 1 paragraph there in a paper, 1896. There's no picture that went with it, so it's kind of hard to see what he was talking about. But there he had a single specimen. There it is. But that's it. And based on that crappy description, you really had to go look at the specimen to see what he was talking about. But that's how it was back then. And in his holotype, he makes a big point at a single specimen. There it is right there. Now, 6 years later, he describes a second species. Let's say it another way. He writes a second name, Animus oxyrhynchus. There's the description. It's a little bit more complete this time. And he gives us a drawing. And I've handled both these specimens. There it is, one specimen. So we have two names. About 15 years later, he takes this species and he puts it in synonymy. That means he erases the name. He says, oh, I was mistaken. This species already existed, this name already existed, and I described it again. So he put his Weeksii in synonymy with another species called maculatus. And no one ever looked at Weeksii again because it didn't exist. It was just another name for this thing. So we have three names here. And when I did my work, there's two species that I can ascribe to these three names. So I've got three names, I've got two evolutionary units, I've got three labels. Which label goes with? That's maculatus, that's the other one. So what you do is you match the names to the types. This Weeksii thing, no one had looked at Weeksii for almost 100 years when I picked this work up. And whatever the holotype is, that's what the name is. That's what the animal is. And the holotype has these black pelvic fins. I know this is interesting, right? And for oxyrhynchus, black pelvic fins. Maculatum, they're clear. That's one of the species distinctions. You can tell them apart that way. So the two with black pelvic fins represent the same evolutionary unit. Those are the labels, those are the types. That's the evolutionary unit. Unfortunately, what Boulanger did is he took this thing and he hid it by saying, oh, it's the same as this. And for 100 years, that's what we thought. And I'm the first person to get all the types out and look at them and realize that this name actually belongs down here, not up here. And you see a problem here? For 100 years, we've been using this name. And this name was hidden. That's the correct name by 6 years. So that was the problem. We've been using this name for 100 years. All the literature had this name in it. That's the correct name by priority. Remember, though, classification is about communication. It's about stability. But it's also the law of priority. And so this is the correct name, which is the name we should start using, but nobody knew this name. This is the name it was familiar with. So to resurrect this name and to bring it out would cause people confusion. So I wrote a paper, and I went to the International Zoological Nomenclatural Commission and said, you know what, this name has been out of use for almost 100 years. Let's just ignore it. We can do that. The rules, if you ask them, they can suppress it. And let's just use this, let's keep using this familiar name. And they said no. So we had to switch to the older name. And many people use this. Main people who are interested in this were aquarium people, but the aquarium books actually fairly quickly started using the correct name. I know, I told you it was exciting, right?
So he just described these two different fish that he thought were different, but then in the end we found out they were the same.
Yeah. He described these two things. He erected two names, thinking he had two different things. And I looked at them and said, nope. The types are the same species. So we have two names for the same species. He unfortunately took this one and said, nope, it's this thing. And then it got hidden for 100 years because no one looked at it again. So it's kind of like detective work. And like I said, we troop back and forth to the library to find fairly obscure papers sometimes. Now so many things are, you just click and it comes up as a PDF. So that, anyway, that's not so much the details are important, but that's the nomenclature, and that's why we have all those rules. I have another exciting example. I think we'll leave it another time. This was another exciting example from Africa, but we'll do it another time. Where I had to match the types, and just before we go, where's the type? The types of one species are all sure. I had to figure out what that was. Well, what it is what the name is, because that's a type. But I had to figure out what what population that represented. Okay. So anyway, the rules just the rules only apply family and down. There's no rules for all this high stuff, other stuff like phyla, kingdom, classes. There's no rules. There's practices, but no rules for up there. In zoology, the rules apply at this level. And by the way, those are the zoological rules. There's another set of rules for botany, and Dr. Dent can tell you about those. Another set of rules for bacteria. But we just play with the zoology ones because I'm a zoologist. So at the end of the day, there's our classification system. And again, this should just be very familiar to you. Kingdom, phylum, class, order, family, genus, species. And it's an information retrieval system. We file things away, and we can retrieve information. Just by knowing that you put a new species in the family Salmonidae, you know something about it already. You know who its closest relatives are, if nothing else. So, orders, in zoology at least, orders, if you see a biology word in your textbook and it's Iformes at the end, that's almost always an order. And not always is an order, and most orders end in Iformes. If it's a suborder, it almost always ends in Oidiae. You know, families like Anabantidae, you see those words almost always end, family names always end in IDEA. Subfamilies always with an N instead of a D and A. And you don't see tribe very often, but tribes are any. So just seeing the word there, you'll know something. And we don't talk. When we're talking taxonomy in this class, we don't usually go down much farther than the order level. All right, let's see. Good? At the end of the day, you remember Stephen Jay Gould, the natural history, evolutionary biology writer? He died actually probably 15, 20 years ago. But he wrote very eloquently, and for the popular reader on natural history, he's got a bunch of books on natural history essays. You should pick them up sometime. But he was at Harvard. Taxonomy, the science of classification, is often undervalued as a glorified form of filing, with each species in its prescribed place in an album. Taxonomy is a fundamental and dynamic science dedicated to exploring the causes and relationships and similarities among organisms. Classifications are theories about the basis of natural order, not dull catalogs merely created to avoid chaos. He defends my science, I find very well. We good? All right, I have, moving on here, let's do this then, we'll probably be good time for a break. Let's see. I've taken things that I think are important out of these two chapters, 8 and 9. We're going to look at some anatomy in lab starting Monday. But I want us to get on the same page with basic animal body construction. And so I need to get some vocabulary out there and some concepts out there. So I call this chapters 8 and 9 bits and pieces. Development, again, remember, to a biologist means embryology. And body plans, well, that's the basic fundamental construction of the body. Vertebrates have a very basic body. If you snake, lizard, fish, mammal, person, line them up, and we have a common body plan. You don't look a lot like a snake, but the underlying pattern is the same. And it's very different than the underlying pattern of a grasshopper or a jellyfish. And that's what we mean by body plans. Why isn't this working? There we go. So there's our evolutionary tree. And in this class, we don't worry much about this because we're only in one phylum. Remember, each of these is a phylum. And we spend all our time in the chordata, where vertebrates are. The later today, we'll define chordates and we'll define vertebrates. I think we'll get that part today. So, as you look at the range of, animals from something like a sponge or an insect compared to a vertebrate or chordate, there's certain things about an insect that are very different than certain things about you. So there's certain things we look at when we talk about body plans. Certain aspects of body plan. And this is the list. By which wondered with me, we did this in a shorter version a long, long time ago. But there's 4 or 5 things we can focus on that are worth focusing on. What do I have there? I have 5 on this list. Tissue, symmetry, germ layers, body cavities, deuterostome, protostome. We'll go through these one by one. Tissues. A tissue, in this context, is a group of the same type of cells that do something. So like a gland, a digestive gland, that'd be tissue, or nervous tissue, or skin tissue. It's a group of one type of cells. And in this jellyfish here, very simple animal, these white structures here, that's digestive gland tissue. Those are masses of digestive gland cells. So that's a tissue. Really simple animals don't have these. If we look at our evolutionary tree here, starting over here, the simplest things, peripheral, those are sponges. They don't have tissues. There's the ancestral animal. Tissues evolve here at the base of all the rest of the animals. So again, as a chordate, we have these. That's a synapomorphy at this level, right? Symmetry is another important one. And again, when we look at the whole animal kingdom, we see variation here. We don't see a lot of variation when we look just at vertebrates. Symmetry comes in 3 types in the animal kingdom. You can have no symmetry. And we spend a lot of time in a zoology class picking on sponges because they're at the very bottom. Sponges have irregular symmetry, which is basically another way of saying no symmetry. The body just grows like a plant. Every sponge is unique. The next step up, a new kind of symmetry. Symmetry evolves, and symmetry is a regular body construction. And the simplest kind of symmetry we still have with us is called radial symmetry. And a jellyfish shows that. Or a sea anemone. Can you picture a sea anemone down Point Magoo? And with radial symmetry, you set that animal down and cut him in half, you get 2 equal sides no matter how you cut him. He's like a pizza. Or a flower pot in this case. There's a sea anemone. Most animals come off the ancestor that invented a more complex kind of symmetry. That's bilateral symmetry. And bilateral, like this lizard here, a tetrapod, means 2 sides or 2 halves. Bilateral. Bilateral symmetry is like this lobster here, or this, continuing the garden analogy, a little shovel. There's only one way you can cut a bilateral animal in half and get 2 equal sides. Bilateral. We have a right and a left. We have a head and a tail. And the key thing here is what evolved here in this ancestor, the ancestor that invented this, is the head. And you know the Greek word for head? Cephalos. Cephalization is when the head evolves. And that gives us, once we have a head, we have a tail, and then we have right and left. So bilateral symmetry is really not about having 2 sides, but having a head. I think they gave it a wrong name almost 200 years ago. So if we look at a more primitive animal, like a jellyfish, no head. That's primitive symmetry. More advanced, we get more sophistication, we get more complex body plans once we have a head. And if you look at our evolutionary tree here again, symmetry, there's our animal ancestor. Well, sponges have no symmetry. Simple symmetry evolves here, and it's still showing in things like jellyfishes, which have radial symmetry. Radial symmetry is probably the first symmetry evolved here, and these guys are still showing it. Bilateral symmetry evolves over here. It came later, and that ancestor gives rise to the rest of the animal kingdom. That was the way to go. And it's that process called cephalization. And what was happening there is animals were starting to move around. This is probably 600 million years ago. And they started to evolve the ability to move in a certain direction, pick a direction, and go in that direction. Oh, there's food over there, I'll go that way. Or there's a predator over here, I'll go away from that. And once animals had the ability to go where they wanted and decide that they wanted to go there, well, a head is an obvious choice. Let's not put it that way. A head is a useful innovation that evolved because you put the control centers up front and you put the sense organs up front. And so as you move into the environment, there's your head. It encounters everything first. Next thing is germ layers. Remember when biologists used the word germ, anyone had development? When biologists use the word germ, that refers to something in the embryo. Biologists never use the word germ as something that makes you sick. That's a TV commercial word. Germ means something to do with the embryo. And germ layers are, think of them, these are starfish embryos. Think of them as sets of stem cells in the embryo. So remember that, we're only in our second day, it seems like the third week. Remember Wednesday, I talked about how animals have a unique embryology? Here it is. This word's not your vocabulary, get it there. The fertilized egg is called the zygote. That zygote starts to divide. And eventually we end up with, in all animals, we end up with an embryo called a blastula. And a blastula is a hollow ball of cells. They just cut this one open so you can see inside. It's a hollow ball of cells. Think tennis ball. Or almost any ball, I guess. And all animals, even sponges, go that far. All other animals, except for sponges, go one step further at least. And that's the formation of separate sets of stem cells. There's only one set of cells here. One set of cells. What happens in everybody else, starting with jellyfishes, is a pocket forms. It's like you took your tennis ball, if you're really, really strong, and could stick your finger into it. Let's think balloon, because you can do that. And a pocket forms. And now we have an embryo with two layers, an outer layer and an inner layer with a space in between. And that embryo is called a gastrula. You looked like that at one point in your life, very early on. And again, these words tell you things. Where are your what's the gastric part of your body?
Stomach.
Your stomach. That should suggest something right there, right? This is the early digestive tract. And it's called gastrulation. And this is probably in a starfish again. There's the blastula. And all this churning and moving around the cells gives rise to that little pocket. And there it is. And once we have that little pocket here, we have two sets of stem cells. We have two, they're called germ layers. One on the inside called endoderm, inside skin, and one on the outside called ectoderm, outside skin. And what those are is those are sets of stem cells. These cells, as the embryo matures, are going to go on and form a specific set of things. They're assigned their job. You're going to become lining of the digestive tract, lining of the lungs, certain digestive structures like parts of the pancreas. That's where those structures come from. These cells are going to go on and divide the ectoderm, and there's your ectoderm right there. But your ectoderm is also your nervous system comes from ectoderm. Parts of your skeletal system start off as ectoderm. Sets of stem cells. I think that's a concept we're pretty familiar with now. The digestive chamber, the pocket I'm calling it, is called the archenteron, which again look at the word there. Arc means old. If you go to a gastroenterologist, what is he or she looking at? Enterologist. Get your digestive tract. Enteron usually refers to intestine, gastric, stomach, enteron, intestine. But there you go. That's the embryonic digestive tract. And there's a hole in it that leads to it. That's called the blastopore. If you have a hole leading to your digestive tract, you say, how many do you have? Holes. Do you have 2? You started with 1, another one forms later. So, there you go. It's just a beautiful little gastro there. Starfish and sea urchins are widely used to study embryology. Question?
So sponges don't have gastrula?
Sponges don't have a gastrula. They have a blastula. They don't have a gastrula. Sponges don't go that far. Okay. Some of this stuff doesn't apply to us because we're talking about vertebrates. We're going to get to vertebrates in a second. But if you only have 2 germ layers, you're called diploblastic. When you see blast in a biology word, it almost always refers to an embryonic cell. And diplo means 2. And things that aren't vertebrates, like jellyfish, are diploblastic. They only have those 2 germ layers. Later in evolution, a 3rd germ layer forms. And it's in this little embryo in red. Again, there's the digestive tract, endoderm, ectoderm. And there's a 3rd layer, and just to continue the pattern of names, it's middle skin, mesoderm. And it's a 3rd set of stem cells. In you, mesoderm is your muscular system, or let's put it another way, your muscular system derives from mesoderm. Your circulatory system, your heart, your veins, your reproductive system, your urogenital system, kidneys, things like that. That's mesoderm. And when you have mesoderm, you're called triploblastic. Okay. Note to self. Let's see what's just going on here. So look at the evolutionary tree again. Oral sponges. None of this applies to sponges. They don't have germ layers. You have to have a gastrula to get to germ layers. They don't do that. So, having 2 germ layers starts here. And again, jellyfishes are still showing that. Almost no one else is. That 3rd germ layer, mesoderm, shows up here. Again, it's the same lineage that gives us bilateral symmetry, gives us germ layers. And everybody else comes off that ancestor. All right, just a couple more things. Let's do one more and then I'm going to get a drink. Body cavities can be confusing. Body cavities, what they are, is they are hollow fluid-filled spaces inside the animal that are not the digestive tract. So these are some worm examples here. See the clear space here? There's the digestive tract. That's the endoderm there. So the digestive tract there. There's a space inside there. These are body cavities. And they come in 3 different types. You can have none, like a really simple worm here. You can have what I'm just going to call a simple body cavity. And you can have a more complex one. I've stopped trying to explain all these different things here. So just none, simple, complex. And what this is this space here, and it gives natural selection another piece of raw material to play with. And we get more complex animals. So things body cavities do for you and have them, one of the important things is there's space inside the animal. Space for organs. This little roundworm here. See all the space here, all the hollowness here? There's the digestive tract, just to orient you. This little worm cut cross section. And all this open space here, and all this stuff here, these are digestive glands, these are reproductive structures. This little guy's not packed full of cells. He's got space for stuff to happen. Organ systems can develop. It's a fluid-filled space. Some animals use it for circulation. They put waste products in there that gases circulate around. You don't have a more complex circulatory system, use that. That was probably a big advantage. Let's see. Why doesn't this little video ever show up? There it is. It could be a structural thing. And here's some little roundworms showing you their body cavity. They're thrashing around. What's happening is that body cavity extends throughout the whole little animal's body, and it's like a water balloon. It's fluid under pressure, and muscles are pulling against it. And so it becomes a skeletal structure. Think of an earthworm. An earthworm doesn't have any bones, but he's, he can get he can pull himself along. Each of his segments in the earthworm's body is packed with fluid, like a water balloon. So, to summarize, once body cavities evolve, they give us a piece of raw material for a more complex body plan. And those three kinds of body cavities, none, simple, complex, are all over the map here. So there's no, what phylogenetic pattern means is, for example, we don't think that one kind of body cavity evolved here, was a synapomorphy for a group, for example, another kind that evolved over here. It's such an important trait. It shows up, disappears, shows up, disappears, shows up, disappears. So it's hard to render a phylogeny based on body cavities. We don't do it anymore. We used to, we don't anymore. So it's a structure that's important evolutionarily, but not in determining classification. Okay. Well, that's about, yeah. Oh, and just the word you're going to see in your textbook, the complex body cavity that you have as vertebrates, what we have is a body cavity called a coelom. So when you see that word there, that means body cavity, and it means the fancy body cavity that we have, the cool one. And that's how it's pronounced, coelom, not colon. The C is soft. So that's not a word I use a whole lot, but you might see it. And by the way, seal, when you see it in a biology word, it means hollow space. If something's hollow, that's often in the word. Okay. Take a minute, put your pencil down for a few minutes, and I'm going to go find a drink and fountain. I've got just one or two more things in this chapter, and then we will move on.
Thank you.
Anyone had ornithology with Dr. Alvarado? You've had
Entomology.
entomology. Anyone entomology? And you've had apiology?
There's an apiculture class.
Apiculture.
And that's that they're offering in the spring.
Apparently, there's a student